I give you Matt. Thanks.
Thanks, Greg. Thanks everybody for coming today. Just very quick microphone check. Can everybody hear me in the room? Yep, more or less. Right. I'll speak a bit louder for David at the back. Yeah. So today I'm going to be presenting some of my PhD work. I'm going to try and squeeze in 2 chapters. The first chapter is looking at savanna community ecology in a small national park in southwestern Angola. And the second part of the talk, which is the bulk of my research, is going to look at the transitions between forests and savannas. And I'm going kind of from a microecological scale to a macroecological scale. But before I do that, I just want to place the research in a bit of context. So I work with forests and savannas in the alternative stable states framework. So this is a theory basically that states that for the same environmental conditions, you can have different vegetation states. And I'm focusing on savannas and forests. They represent over 60% of land surface in Africa. They're the 2 biomes that deliver the most important ecological services. And basically, you have these feedback mechanisms. So for example, in savannas, you have grasses which catch fire, which reduce tree cover, which promote more grasses, etc., etc. Whereas in forests, you have trees that create shade, eliminate grasses, eliminate fire, which creates more space for trees, which creates more shade, etc., etc. And so these 2 systems are kind of adapted to opposite ends of the ecological spectrum. And in order to switch from one to the other, you kind of have to climb this ecological hill. And anything that's in between is unstable, basically. So I'll move on to the first part of the talk, which looks at community savanna ecology in Bikwa National Park. So this is a national park in southwestern Angola. On the right-hand side, you can see some images of what the vegetation looks like. So on the first image here, I'm not sure if I can zoom in. Yeah, you have one of our colleagues, Luisa, here at the bottom, and some of the trees. So you can see how tall it gets. Pretty open ecosystem. On the bottom, whoops, sorry. The bottom image, you can see maybe it gets a bit denser. So that's, for example, where a fire has occurred and there's lots of regenerations of the stems. On the right-hand side, I've gone and put a satellite imagery of the different plots that we have in Bikwa, where they're located. And there's a faint layer where you can basically see the currents of fire in the system. And so fire is thought to be a really important driver of these systems. It's a rejuvenator. It's a driver of diversity. So some of the questions that I'm interested in are, what are the main drivers of species composition in Bikwa? What is the relative influence of environmental filtering versus competition versus neutral processes? And what explains the co-occurrence of species? We find that some species never grow together, some species always grow together. So what might explain that? So the first thing I'm doing here is building an ordination. And so this is a type of redundancy analysis. So you can see that, sorry, I'm not getting the laser to work here, unfortunately. So I'll just describe it. So the blue arrows and the blue letters correspond to the environmental predictors. The one on the right hand side, which says AVE, is basically a predictor of topography. So that's what we call the catenal position. So catena is basically like a slope. So it's either you're at the top of the slope or at the bottom of the slope. The names in red are the species and the letters in black are the different plots. And so you can see here that fire hasn't been selected by our model, but most of the variance is explained by either the catenal position or the soil variables. And the model explains about half of the variance within the data, which is quite good. If we partition the variance between the different predictors, so on the left-hand side, I've gone and categorized my predictors as either soil variables or fire variables or catenal position. What you can see here is that fire, which is in red, doesn't actually explain any unique variance in the data. Most of the variance can be explained only by soils and a combination of soils and catenal position, which is pretty strange for a savanna ecosystem. This is not something that I was expecting at all. On the right-hand side, I've gone and taken out only the soil variables, and I'm doing the same exercise of variance partitioning, but just between 4 of the major soil variables. And you can see that the CN ratio in the ground is what's explaining the most variance out of the soil variables. The next thing I'm doing is using a joint species distribution model. And so here we're able not only to get global scores, but we're able to get species specific scores from our model. So what I'm showing on the left-hand side here is basically which of the environmental predictors are affecting species individually. So on the top here, you can see Baikia plurijuga has a strong positive association with catena position. That means that we find that they grow mainly at the top of the catena, whereas right at the bottom of the graph, Bokia africana is heavily negatively affected by the impact of magnesium in the soil, for example. So with that, you can kind of build a species-specific environmental niche for all of our species. And you can do the same exercise as before. You can partition the variance. Here it's between the environment. We can include spatial predictors. So I've gone and included Moran's eigenvector maps as spatial predictors of my data. And the model also pulls out the biotic associations. So that's the amount of covariance which is only explained by the relationship between the species without the environment and without the space involved. And you can go a step further. You can decompose that for each of the sites and each of the species separately. So this is called the internal meta-community structure. And you can see that environmental filtering is the agent which is playing the most heavily in the composition of the system. The final part, which I'm interested in, so we've got our environmental niche parameters, and I'm also interested in how much of the community composition can be explained by its functional traits or its phylogenetic affinities. So do species tend to co-occur because they're more closely related to each other in the phylogeny, or do they tend to co-occur because they have very different functional niches? So what I've done here is I've built a phylogenetic PCA with my functional traits, and I've extracted the first two axes of the ordination, which explain the most variance. And I've gone and color-coded them according to the trait values and plotted them back onto the phylogeny. So what this shows is that closely related species don't tend to have similar traits. So they have very different functional strategies, basically. And so once we have all of that, we can look at which of these different facets of diversity explain the co-occurrence of species. Is it because they occupy different functional niches? Is it because they're closely related? Or is it because they have very different environmental preferences? And so I'm using simple mental tests for this. So I look at the co-occurrence of species versus their functional distance. And unfortunately, this is not significant. There doesn't seem to be a relationship here. I do the same thing with phylogenetic distance, but again, this is non-significant. What is significant is the ecological distances. So the ecological niche that species prefer is really what's driving the co-occurrence of species. And this kind of makes sense given the results that we found so far with the environmental filtering. We can see that there's low amounts of competition in the system. And so perhaps species don't necessarily need to be functionally different from one another. So to summarize all of this, we can see that environmental filtering is what's strongly shaping the community composition in Bikwa. This is acting through the position of species along the catena and their preferences for soil, in particular CN ratio. And we're seeing that fire doesn't explain the community composition here, which is a really important finding. And the second aspect of species co-occurrence here is that, as I mentioned, environmental parameters are really what are driving the co-occurrence of species in this system, not functional distance, not phylogenetic relationships. Great. So now I'm going to move on to the second part of the talk, and we're scaling up here. We're looking at the whole continent, and I'm showing a figure here from a paper that came out a couple of years ago from Julie Allemand and colleagues back in 2020. And what they did is they basically took a bunch of sites. Those are the crosses that you can see on the maps. And they took species lists from those sites, and based on that, they were able to pull out which species corresponded to forest plots or savanna plots, and what is intermediate. And so this map shows basically in dark green, we have the core forest area, which is the Congo Basin and Western Guinea Congolia. In cream color, you have like the core savanna zone. And then in light green and in orange, you have these bistable states where there's fluctuation. You can be either a forest, either a savanna, but with some sort of preference. And so the questions that I'm interested in here are regarding alternative stable states. So from plot data, can we figure out a signal of alternative stable states? What can floristics versus structure tell us about our systems? When I say structure here, I mean, for example, the number of trees in a plot, or the height of the trees in a plot, or the standard deviation of the diameters of the trees. So something that's completely unrelated to floristics. And finally, are there any intermediate states that are possible between forests and savannas, and are they stable at all? So to look at this, I've gone and built some maps here. On the left-hand side, you can see a bunch where all of our plots are located in Africa. So those are the colored points. I've gone and color-coded them according to a biome index. So the way that I've done this is that for each plot, I've pulled out all of the species, and each species can be assigned a biome preference. Is it a a forest species, or is it a savanna species? And you basically, for each plot, you get a ratio of forest to savanna species. So that's a continuous index. And you can see that most of the plots that are in green, the forest plots, they occur where we know they are in the Congo Basin in West Africa. And most of the savanna plots occur here in South and Eastern Africa. Now, you'll notice that along the scale, there is a blue line. That's the intermediate plots. And you can see that there aren't that many. There are a couple which are located mainly in East Africa, here in Tanzania. But there are others as well in West Africa that you can't see because of the way that I've arranged the points. On the right-hand side is a map of herbarium data. So this is georeferenced, expert identified material that I've gone and plotted on Africa. And essentially what I'm doing here is I want to find a way how to combine plot data with herbarium data. So in order to do that, I've selected my plots, and around each of the plots I've created a buffer zone. So this buffer zone can be the size that you want. I've decided that it would be 100 kilometers. And essentially that's going to represent the species pool around the plot from which it can recruit different species. And those will be from the herbarium data. So this allows us to get some sort of expectation of what the species pool is around a plot. And so with that, you can build simple binomial tests. So this is where you would get the occurrence. Oops, sorry. You would get the occurrence of a particular type of biome observation. So that's like the number of forest species that are in your plot versus the total amount of stems in the plot, given the expectation of the forest biome from the herbarium data. So what this tells us is that on average, there are less transitional plots than we would expect given the herbarium data. The plots that are forest or that are savanna, they're actually more forest and more savanna-like than we would expect given the herbarium data. And finally, we can see that if the species pool around you is forest, then there's only 4 times where you get a savanna plot. And vice versa, if the species pool around you is entirely composed of savanna species, you never get a forest plot. So what this is telling us is that the species pool has a very strong impact on what you're going to be as a vegetation state. But there's some sort of force that's pushing you to be more extreme than you would expect given the species pool. So perhaps this is telling us something about alternative stable states. Okay, the next part is looking at how floristics and structure can tell us what's happening in our systems. So it's a bit of a mouthful, but basically what I'm trying to say here is that you can either let the data speak for itself, so that's a completely unsupervised way of looking at the data, patterns emerge from the data without you controlling for anything. Or you can come at it with an a priori expectation. You can ask specific questions from the data given a pattern that you expect. And so that pattern might be the continuous variable, which I presented on the first slide, which is like the biome index, it varies along the continuous axis. Or you can decide to make that a categorical variable, either forest, either savanna. But then you have to decide what the threshold is between the two. So I'm using a bit of both for visualization purposes. So the first thing I'm doing is I'm building an evolutionary ordination. And so this is looking at where plots group together based on their species composition. So you can see here that the forest plots, which I've colored in green, they all seem to group together nicely on the right-hand side of the graph. But the savanna plots, they seem to be more diffuse along the second axis here. And that's all of the plots in southern Africa, which are located here in the bottom left of the graph, and all of the plots in mesic dystrophic savannas, so that's more western, northern savannas that are separating out. I can do the same thing looking at my plots in structural space. So I've gone and pulled out some structural metrics like the number of trees per hectare, how many stems can a tree have. And again, here you seem to have some sort of very strong pattern for being a forest or a savanna structurally. But there are some plots which seem to be a little bit in the middle. So from this, looking at floristics and structure separately, you can extract the axes from these ordinations. And with these axes, you can do some sort of clustering. So you could do a hierarchical clustering, or in this case, I'm deciding to use normal mixture models for clustering. So the output of that is basically you are either a forest, either a savanna, But the benefit of using these types of models is they give you some sort of uncertainty measure. So you can say, well, if it's a bit in the middle, we think it's this, but we're not too sure. So from the clustering, I'm basically building a confusion matrix, and I'm looking at when the two types of clusterings agree with each other. So that would be in this box here, for example, both the flouristics and the structure agree that these plots are forests, and vice versa in this box down here, sorry, this one here, both the flouristics and the structure agree that these plots are savanna. So that's about 80% of the plots where flouristics and structure agree together. That's quite nice. But that still leaves us with about 20% of the data, which there's some sort of disagreement. Either there's uncertainty or there's clear disagreement. For example, here, there's at least 35 plots for which the structure says this is definitely a savanna, but the flouristics say, no, this is a forest. And then up here, you have at least 6 plots where the flouristics say this is for sure a savanna, but the structure says, no, this is definitely a forest. So I've gone and plotted them on a map of Africa to see if there's some sort of geographic signal here. And at least for these ones, so this is the uncertain structural plots, there's quite a lot of uncertainty. And these seem to be grouped along the Congo Basin mainly. And the plots where they disagree here seem to be mainly in West Africa and a little bit in southern Congo here. And if you go back to the metadata of the plots, you can actually see that, well, these seem to be logged forests. So there's some sort of disturbance. There's less trees than you would expect, smaller diameters, even though most of the tree species are forests. So this is giving us a nice little pattern and way of detecting like disturbance in these systems. Okay, so far we've looked at how flouristics and structure can tell us things separately, but what if we could combine them together? So if we go back to our ordinations, we can turn these into dissimilarity matrices, and then from this, we can simply average the two dissimilarity matrices to get one combined matrix. And now from this, that's great. You can do some modeling. You can use it directly for generalized dissimilarity modeling, or you can do some more clustering with this, either hierarchical or, again, some Gaussian mixture models, which I'm going to show you. So this is the output. You have two clusters, either forest, either savanna, and the plots that are in the points that are in red on the right-hand graph are the ones where there's a lot of uncertainty. We're not too sure if they're forest or savanna. One more minute. Okay, sorry, I'm going to speed through this because there's a bit left. And basically what we're seeing here is that from our expectations, given the biome index, most of the data seems to agree. Most of the data says, oh yeah, this clustering is really doing a good job. There's only a few points which are savannas, which are kind of grouping in floristic space. But on the whole, it's not too bad. I might skip this very quickly because I don't have much time left. But basically, this method is a way of combining lots of different types of information, spatial information, floristic information, phylogenetic information, and it comes up with some sort of global ordination. And on that, you can plot your environmental predictors. You can see which species scores along the phylogeny you have. So it seems to be a very strong signal here for being either a forest or a savanna. And this is how they group on a map of Africa. So in this way, you can see from a completely unsupervised method, we're getting something which is approximating our expectations quite a lot, even though on this map, there seems to be a lot more intermediate states possible. The very final bit of the talk, which I'm going to whiz through, is looking at diameter distributions. And so from this, I've selected the plots which are intermediate between forests and savannas. And usually we can get a measure of how disturbed a site is based on how steep the diameter distribution is. If you have a nicely smooth gradient of diameters, you can think that this is a relatively healthy and undisturbed system. Whereas if there's a very steep curve, that means there's lots of disturbance in the system. So I'm going to focus on 3 plots here, where you can see a nice gradual decline of diameter distributions. And in these plots, you seem to have an equal amount of forest trees and savanna trees. And these plots, they're grouped both in floristic space, they're intermediate, and in structural space, they're intermediate. So this is perhaps telling us that intermediate states are possible, and maybe there's signs of stability here that are going on. So just to conclude, can we detect a signal of alternative stable states from the plot data? So using these binomial tests, we can see that there's less transitional sites than expected given the herbarium data, and that plots tend to be more extreme than you would expect. They're either more strongly forest or more strongly savanna. The flouristics and the structure, they agree over 80% of the time, and that supervised and unsupervised way of looking at the data kind of converge most of the time. And we can see from the environmental predictors that variables that are linked to water and temperature and soil are the most important for predicting our vegetation states, and that fire isn't. And finally, are there any intermediate states that are possible? Well, yes. We know that some are possible. We can detect it from our plot data. They can be intermediate in both their flouristics, intermediate structurally, and there seems to be some signs that perhaps some of them can even be stable in this state. So yeah, that's it. Sorry for going over time.
Great, many thanks indeed. And folks, I wonder if anyone has. They do indeed have questions. I'll bring this mic round, and would you be okay just to sort of repeat the question just for the folks online?
Yeah, no problem.
Thanks, Matt. That was awesome. So the first bit of your talk, you said that co-occurring, at the end you said co-occurring species were ecologically similar, and so there wasn't much competition. Why do you think there's not much competition? Was it like succession habitats as a disturbance?
Yeah, so the question is about how species co-occur if there's an effective disturbance in the system. So from the joint species distribution modeling, what I'm finding is that competition doesn't play a strong role in the system. And so that means that you can basically go anywhere and you're not going to find that many other individuals to compete with. And so if that's the case, you're kind of free in your functional strategies to basically occupy the land however you wish. But that you're still constrained by some fundamental environmental preference. So each of the species, they like to grow in certain environments, and they will favor those environments regardless of which other species are present or not. That's how I interpret it.